Doug he has two bachelor's degrees, really fascinating background. Dr. Wacker, you have a bachelor's degree in criminal justice and BA in telecommunications, and then you have a BS in biological sciences. But he's a Husky, which means he received his Ph.D. in neurobiology and behavior from University of Washington, Seattle. And his research is in the behavior of free-living vertebrates. I'm liberally quoting from his website. And essentially, he's looking into learning how circulating hormones and the brain interact to alter social behavior in birds. He's also been pursuing pure behavioral studies, and we can go to his website to learn more about his research. Of course, if you're interested in crows on campus, I would strongly recommend check out his work, because he's been doing some fascinating work with the crows, which has kind of become a symbolic issue on campus, because, we are a campus where we have a lot of crows. I'm sure students who have been on campus, especially during the sunset time, it's spectacular. So today, Dr. Wacker is going to talk about his project on the Lake Forest Park Community Bird Project, assessing the long-term impacts of urbanization on bird species, diversity, and behavior. So I'm going to hand it over to Dr. Wacker. Dr. Wacker, the Zoom floor is yours. Please take us on the journey.
All right. Well, first off, thanks for a great talk, Dr. Madzima. Fascinating to hear about the work that you're doing with the SRCP funds and about your student involvement. So thanks for that. And thanks everybody for sticking around and listening to what we're up to for the Lake Forest Park Community Bird Project. And this project was enabled by funds through the SRCP seed grant. So I'm definitely very appreciative of you to Bothell for offering me this opportunity to do this research. And we're endeavoring to assess the long-term impacts of changing habitat and urbanization on bird species diversity and behavior in green spaces and parks in the Seattle metropolitan area. And the goal of this project is really a long-term one. It's to look within individual sites over a long time, even years, and to see how changes in urbanization and habitat structure interact to kind of are reflected in changes within sites over a long period of time. And I have collected 1 year, myself and undergraduates, I'll mention in a moment, I've collected data for about 1 year. So of course we can't look at one site over a long period of time yet. So the results that I'll talk about today are preliminary and fairly descriptive. But I'd be excited to come back in like 10 or 20 years and tell you what we found in the long term. So, and I could not have done this work without the involvement of some undergraduates. We can see Andrea Castro here, Kira Lemke, who designed this logo for our project with the Sellers J, and Alex Mosley, and I'll talk about their involvement as we kind of go on here. So first thing I want to do is just say, why are we interested in looking at bird species diversity, the number of species, how many birds of each species are there, how evenly distributed are species and populations, and behavior in the Seattle metropolitan area? Why are we even interested in that? Well, this paper came out in 2019. And I wanted to put the abstract up because you should read the abstract and you should definitely read the paper. I'd be happy to give you a copy of it. It's called The Decline of North American Avifauna. And I want to just highlight one sentence in this abstract, which is crazy, really, if you read it. An integration of range-wide population trajectories and size estimates indicates a net loss approaching 3 billion birds, or 29% of the total abundance in 1970. That is an astronomical decline in bird numbers. And we really need to figure out why we've seen this and potentially are continuing to see this decline in bird numbers, because this will eventually lead to extinction of bird species, right? So there are a lot of reasons for this. And one reason may be that humans and their impact has changed over over time of the last few decades. And we see this through increased urbanization and densification of our areas. And anybody that has lived close to Seattle will be familiar with what I'm going to show you in this next video. So this is a video that was created by Ricardo Martin Barala. His website's here. This is a great little site here on medium.com about how he designed this time-lapse series of videos taken from atop the Space Needle from 2015 through 2017, looking at how Seattle is changing. All right, so I want to show you this video now. So we can see as time goes on, more and more buildings, more pavement. This is urbanization. Big buildings, right? Big residential condos, densification, more and more people in this very limited space. But we also see some green space. We can see these trees down here in a park, right? And that's still there. And there are birds and other animals and urban wildlife that utilize that green space. How are they being affected by this kind of radical change that's going on around them? And that's the question that we're trying to begin to answer, partially through this project that I'm going to describe. The video actually that Ricardo created is much longer, and it's definitely worth checking out the whole thing. It's pretty amazing. Okay, so this is the Lake Forest Park Community Bird Project, not the Seattle Community Bird Project. So where is Lake Forest Park and why is it important? So Lake Forest Park is a suburban community just north of Seattle, north of Lake Washington. And it is a community that has had a history of environmental stewardship and activism. In fact, when Lake Forest Park was founded by Ole Hansen in 1912, he said, "Here the laughing waters will forever make gladsome the hearts of the sylvan dwellers." This was supposed to be a location where people could live within the natural environment of the Pacific Northwest. And can really highlight this kind of environmental stewardship and really environmental activism by looking at one little area of Lake Forest Park in this little square. Zoom in here. This is a little triangular area that is separated by 522. You might take that to work at UW-Bothell if you work at UW-Bothell, and Ballinger Way, which goes to I-5. And this is what this area looked like in 1936, a wooded wetland area. And in the 1950s, business leaders in Lake Forest Park wanted to convert this area into a shopping center. And the community rallied together and said, "No way, we want to have our trees and our water. We don't want you to build here." And they incorporated Lake Forest Park into a town, and it didn't matter. The shopping center, even though they were incorporated in 1961, the shopping center still went up in 1964. It's called the town center. And this is what that region looks like now. These are aerial photographs from King County IMAP. So you can see it looks quite different. It's quite developed. So we can take a closer look at this. This area is low-rise commercial. It's basically strip malls. Kim Wacker, my wife, took these photos. Thanks, Kim, for these photos the other day. And you can see what it looks like. It's developed. But what's interesting is right next to this town center, there are a number of parks and green spaces in important environmental areas. For instance, there's Whispering Willow Park and Lying Creek Park and Preserve. And Lying Creek is a salmon-bearing stream, and right across the street, Lying Creek goes into Lake Washington. So these are really environmentally sensitive areas right next to this development. So in 2019, the owners of the town center in Lake Forest Park proposed some very radical development of this area yet again. And this is from the draft environmental impact statement of what they wanted to do. They wanted to build these kind of big multi-use, partially residential, partially commercial condominiums on the site, over 65 feet tall, right next to, again, these salmon-bearing streams and parks and green spaces. And we really wanted to take the opportunity to see how this development might impact these nearby green spaces. So that's why we picked Lake Forest Park. Now, this development's not going to look like this anymore because, again, the citizens of Lake Forest Park came out and kind of said, no, this is inconsistent with our environmental vision for this community, but there will be development there. So we want to follow and see what the impacts are on bird species diversity and behavior in these areas. Okay. So where are we doing this work? Well, part of our sample is in Lake Forest Park. We have 11 parks and green spaces from which we conduct bird surveys, again, looking at species and the behavior of these species. But we also have 4 sites in northern Seattle. Each site is one of these little birds in a green circle. These are arguably more urban sites because we're interested in urbanization, but we'll come back to that, as well as some sampled sites in rural King County and some sites in Snoqualmie, which are more newly developed. Right? So we have some kind of rural and urban companion sites to take a look at. And what have we done this far? Oh, by the way, I'm going to go back. I want to point out, I'm going to have some photos in this talk of birds from Lake Forest Park taken by folks that live in Lake Forest Park. So Lynn Hakeem is a LFP resident, took a picture of this house finch, and I'll point out these photos as we go along. This is an Annis hummingbird, photo taken by Maggie Forrester, also LFP resident. So what have we done this far? Well, we've completed an urbanization analysis, and I'll talk about that in a moment, looking at urbanization in each of our areas. We've looked at and done kind of a general habitat assessment for each of the areas we're going to look at, and as well as an edge analysis, and I'll explain what that is and why it's important in a moment. We've conducted 132 surveys across 33 sites and 24 parks, and we've done this across 4 seasons. The seasons are important because they correspond with different life history stages of the birds that we're interested in. There are birds that are only here, for instance, in spring and summer, and then migrate south for the winter and fall. So we're going to see different bird species depending on the time of year that we're looking at. So we do these surveys across different seasons. And we've begun a preliminary analysis. But remember, our goal here is to examine each site over years as urbanization and habitat patterns change. We're really interested in diversity and behavior within these sites rather than across them. But we have a year's data, so we're going to look a little bit across sites today. And then finally, we've started to forge a community engagement component of this project, which is really near and dear to my heart, which I'll talk a little bit about at the end of the presentation. Okay, so let's talk about each of these things. We'll talk about the urbanization analysis first. How do we determine how urban an area is? Well, we get an aerial photograph of each location, and we lay a 1 kilometer grid. There's 10 cells in each row, in each column of this grid. And then we zoom into these cells, and we look at a variety of metrics of urbanization. So we look at buildings, we look at paved surfaces, and we look at vegetation. And then we use a statistical technique called principal component analyses to kind of combine all of these different variables into one variable we call urbanization. And if you know anything about principal component analysis, you might be interested in what are called the factor loadings for that analysis, basically how much and which direction they contribute to this urbanization value. And we can see that things like buildings and pavement fall out together while vegetation falls out in another direction. So that's good. We know that our urbanization analysis is sound. And this is based on pre-existing work by Sarah Settle in 2014, which has been established as a good way to look at urbanization. So that's our urbanization analysis. What about habitat analyses? We go out with students in our GPS units and we figure out what's there. And this is pretty crude, and we'll talk about that in a bit too. But we can see this is Sphinx Animal Acres Park, and we can see, 53% of this park is trees, whereas a small part is completely developed. It's just pavement, parking spaces. We've got some area here that's predominantly shrub covered, and we can use this information to figure out how habitat might change or interact with urbanization. We also do, we've done this for each of the parks and green spaces in our study. We also do what's called an edge analysis. And this is looking at the habitat that's just on the edge of the parks and green spaces. And we know this is important because the animals don't just live in the park and never leave. What's right next to them can really impact what species you see and how they use the surrounding environment. So what we can see here, this is also Animal Acres Park. It's a little harder to see, but some of these areas of the park are bordered by more trees. Others are bordered by kind of mixed habitat residential, so houses with yards and trees and driveways and so forth. And then some of these sides are bordered by road, and I'm not showing you here, but anytime there's a road on the edge, we look at what's just beyond it, and we also kind of examine that because that might be important as well. Okay, what about the bird surveys themselves, right? What about counting the species and so forth? What do we do there? Well, for our bird surveys, we start at sunrise, and we end within 3 hours of sunrise. And we do what are called point counts. So that's Andrea Castro and Alex Mosley, some students that work with me. I always go out with them as well. So I'm the kind of common factor across all these shifting undergrads, which, you know, we're not gonna have the same undergrads hopefully 10 years from now that we do now. So those are going to shift, but I'm going to be the common factor here. And they determine about what's 50 meters away. We do a lot of practice there, and we kind of make an imaginary cylinder over the top of that. And within this 50-meter radius cylinder, we record all the species of birds that we see and hear for a 10-minute period. Okay. It's consistent with what is done in a lot of these studies, so we can compare this across other scholarly work. We also note the behavior and habitat of all the birds that we identify as possible, and I'll talk about that in a sec. And we record temperature, wind speed, relative humidity, noise, and other abiotic variables. So when we look at habitat, how do we do that for, you know, how the bird is interacting with its habitat? If this is a nice northern flicker taken by Lynn on some developed habitat there. We just have codes, and the students and I learn these codes before we go out. There's a bit of training. And, you know, if the bird's flying, it's in the air. If it's in a tree, we kind of determine where it is in the tree. And this can give us some information about how particular bird species are utilizing these parks and green spaces. And then we also look at the behavior that we witness. There's a lot here. I'm not going to go through each line of this, of course. But I do want to point out this area down here for sounds. That'll become really important. They can make calls, they can sing. The difference between songs and calls, I can certainly get into that later if somebody's interested. And that we found is a really important thing to know. Excuse me. Okay, so we monitor how many birds we see of each species, where they're at, what they're doing, and then how do we pull kind of biodiversity measurements out of that information? Well, in order to explain that, I want to give a little biodiversity 101. These are drawings that Kira Lemke did. It was an undergraduate that worked with me. And we can see two populations of birds here. Perhaps these are birds that we saw on two different surveys. We can see that both in the group on the left and the right, there are 5 species. I see Steller's jay, American goldfinch, Anna's hummingbird, pileated woodpecker, and some kind of dabbling duck that I'd be able to identify as soon as it was done dabbling. So same, 5 species in each side here. 10 total birds in each of these groups too, okay? But these groups are clearly different because in the group on the right, that's dominated by these American goldfinches. So we have an uneven distribution of species on the right and a more even distribution on the left. So here we're looking at species richness, the number of species abundance, how many of each animal is in each species that we see, and then evenness, how evenly distributed they are kind of across a particular group. So that's how we're measuring biodiversity. What have we seen thus far? Okay, so again, fairly preliminary data here, but I'm going to show you what we found thus far. So year one species abundance. We have identified over 3,400 individual birds, which is great fun for someone who enjoys bird watching and be able to do this for a living. That was wonderful. We identified over 50 different species just in our surveys alone. Now, when we were practicing, we saw additional species as well. But still, I think this is kind of amazing for folks that don't think about birds that much, that, wow, you can go out to these places and see 50 different species of bird. So it's much more diverse than I think kind of a non-birder would appreciate. And 67% of the birds that we identified were identified by sound alone. My PI in graduate school, John Wingfield, used to tell me that 90% of bird watching was listening. And I wish I would have listened to him a little bit more closely because I was not prepared for this. And some of these sounds in the breeding season, these birds have diverged to sing quite different songs because they're very important, they're species specific. But in the non-breeding period, there's a lot of convergence and a lot of these sounds sound very similar. And it took a lot of time for my undergraduates and I to work together to be able to differentiate some of these very similar sounds in the field and be able to identify some of these birds by sound alone. So what have we seen? It's going to be a little hard to read this, but I wanted to include all of them. These are the birds that we've identified. And some of them, we only saw a couple. We saw Bullock's oriole in Quigley Park in Falls City. That was really cool, eating from some kind of ornamental cherry plant. And then other birds like the American crow, we just saw or heard a whole lot of, right? So these are the birds that we identified more than 25 over the course of the year. And again, we can see that the American crow was the one that we saw the most or heard the most. Followed by the pine siskin. This is an interesting story. There was an eruption of pine siskins last year. So they are an eruptive migrant. They move erratically, so not predictably south and some years and not other years. And last year we had a big group of pine siskins move in. And it was a real problem, actually. You may have heard in the news that you should take your bird feeders down last year because there was a salmonella outbreak. And that was because of this influx of pine siskins. And in fact, a few people in Washington got sick from that, from handling their bird feeder and not washing their hands before they ate or something. So lots of pine siskins, we may see this number go down. That'll be important too when I show you a couple other graphs coming up. So lots of birds we saw. That's kind of an abundance thing. Let's look at species. This is a graph showing the number of species identified, the average per survey, these 10-minute surveys by season. And we see that there are more species seen or heard in spring and summer. This makes sense because, again, we have a whole load of migrants that are here in the spring and summer that leave in autumn and winter. So this, not incredibly surprising, but made us feel pretty good about what we were able to identify and not identify in the field. This pattern was expected. Okay, what about species richness by urbanization? Interesting, I'm interested in urbanization. Now, again, I really need to look at these sites individually over time. There's power in that. I'll talk about that in a second. But if we just look at all the different sites in each season, and on the x-axis, we have that urbanization score that integrates all that thing, building, pavement, vegetation, right? And on the y-axis, we have the number of species that we see. We don't see a pattern between urbanization and species richness. But honestly, it's not surprising. I just finished entering some of these data last week. So we have not been able to integrate, the habitat, the edge analyses, anthropogenic noise, which we think makes a big difference here. So we're missing some things from this analysis thus far, and I look forward to kind of integrating that as the year goes on. Also, there may be hidden variables that are important that we're not considering, and that's really the power of looking at these individual sites over time and kind of a repeated measures approach. Animal Acres Park might have some differences from Grace Cole Park or Horizon View Park. They look very similar that we can't recognize. So by comparing Animal Acres to itself over years, we kind of control for those without even necessarily knowing what all those variables are. So that's a power here, and why we may not be seeing a difference. What about species evenness? Like how evenly distributed those species are across these different urbanization values. Again, we don't see a relationship with urbanization on species evenness. There's some interesting things here to show in these graphs, though. There are some dots here, some points. Each one of these is a location, so a survey. Actually, it's a location. There are some sites that we survey more than once, and then we average everything because the sites are larger than some of the others, so to control for size. But there are some spots underneath the line here, and we think that those lower evenness scores in winter and autumn, but not necessarily in spring, are due to some of those flocks of pine siskins, American goldfinches that we see in those seasons, but don't see during the breeding season. But nonetheless, even though I'm not worried that we didn't see any patterns here yet, it did get us thinking about the way that we're measuring urbanization. So I want to look back at that. Here are our sites. Here's how they fall from more urban to less urban. I'm going to highlight the sites in Lake Forest Park. They kind of fall across the spectrum a little bit. You know, one might expect with a suburb that they might kind of pull out in the middle a little bit somewhere, right? But, you know, let's look at the Seattle sites, the more urban sites, arguably. Well, they fall out as more urban, just like we would expect. The blue sites at the top, those are the sites in Seattle proper. But what about these rural sites out here in Eastern Washington, right? Or Eastern Washington, Eastern Western Washington, Eastern King County. Those are in green. They kind of fall all across the board there. Some of them are more urban, some are less urban. And we think the reason for this is that, you know, in some of these rural areas, they're quite dense where there is housing, and then you kind of have fields, right? So we need to capture that somehow. And also Lake Forest Park is known for their, again, for their environmental stewardship, for their protection of their tree canopy. There are really strict laws about cutting trees of a certain diameter at breast height. You have to go through a big approval process. So we got a lot of trees in Lake Forest Park. In fact, they fall down. I live in Lake Forest Park, and the trees fall down a lot in winter with winter storms. And Seattle City Light calls us Lake Forest Dark because we have so many power outages, because we have so many trees that go down. But we have a lot of trees, and, you know, an aerial photograph, some of those buildings and pavement might be obscured, right? So what are we doing now? Well, one thing that we're doing is we're trying to find new ways to quantify that urbanization, because we might be missing some of this because of the tree canopy, because of some of this kind of denseness, lack of denseness in the rural areas. We're actually going to start measuring canopy cover on these sites, because if that's important, we want to know, right? We want to see how that changes with urbanization. I'm seeking a collaborator and to enable a more fine-grained habitat analysis, including some plant inventories at some of these sites, which is going to be more important than just saying trees and grass, right? It's going to give us a better idea of what might be going in, and maybe how native plants could be affecting what birds we see in these areas as well. And finally, we're integrating citizen scientists into our project via eBird surveys. And I want to briefly talk about that for an end here. I got a small grant from an outreach grant from the Animal Behavior Society to hire UW Bothell undergrad Wyatt Olson to create a website for a public-facing website for our project. It's not live yet. It's out there, but it's hidden. It's not live yet. Once it's live, I'll share a link. And this will not only involve the community, which is really a pillar of what we expect here at UW Bothell, but it will help us with our research, because we're going to get citizen scientists to do some surveys themselves. And we can use an app that was already designed by the Cornell Lab of Ornithology to help us with this. It's called eBird. Folks can go out to these sites. They have an app on their phone. They hit go, and it times them. It tracks how they walk, so they don't have to do a point survey. They can walk through these areas, and they record not behavior and habitat, but every bird they see or hear. And then we can do a couple of things with these data. I can get these data from Cornell. I've started to do that already. We can see if maybe if we get a lot of people out there doing surveys, will they identify some birds we didn't see, maybe some rare species we don't see in our shorter surveys. And then from a scholarly point of view, we can compare the findings of our systematic surveys to these public surveys and see what's different. And that will help us moving forward. Okay, so with that, I'd like to thank you all for listening. I definitely want to thank you to Bothell, SRCP, Seed Grant, and Lake Forest Park Stewardship Foundation and ABS for funding for this project. Lake Forest Park Stewardship Foundation is a not-for-profit in Lake Forest Park that's been very supportive of this research. Dan Bragg, David Hepp, and Don Norman for bird identification help. Don Norman's Puget Sound Bird Observatory and gave me some great tips for differentiating non-breeding bird calls. Local photographers for the beautiful bird photos, and then most importantly, the undergraduates down here, Alex Mosley, Kira Lemke, and Andrea Castro, Wyatt Olson, Maya Hahn, and Brian Hutt, who have gone out and done all these surveys with me and helped me collect all the data that you've seen here today. So I couldn't do that work without them. So with that, thank you, and I'd be happy to answer any questions.
Well, thank you, Doug. That was fascinating. Ornithology has been a long-time hobby of mine, so this was just gratifying from that perspective. But I think the study itself is really. I think you guys are starting to scratch the tip of the iceberg, as they say, the proverb. There's just so much richness to this study, and I think I'll be here to keep you company over the next 10 years. I look forward to seeing what you find, because this is really amazing work. Let's open the floor for questions. Let's invite our audience to ask you some questions.
Yeah, Laren, I see you have your hand up. Dr. Lewis.
I always have questions. So my question for you is, you mentioned that in winter and autumn, you thought that some of the numbers had been changed due to these sort of huge run-throughs of a bird that honestly I don't even know that I would recognize if it landed on me. But my question is, can you disaggregate that information from the rest of your data, and do you see those things go away if you remove those?
Yeah, so
trends go away, or
yeah. So again, well, I mean, there's no trend anyway. So it just happens to be some spots underneath the curve, and I was literally entering a lot of this data last week, so I haven't looked carefully at it. But some of those data points are definitely those flocking birds, and it's not just the eruptive migrants, but birds tend to be more. Birds are amazing. The birds here are seasonal breeders. I go into endocrine mode a little bit. They basically, at the breeding season, they grow their gonads, their testes or ovaries, and they become in breeding condition. At the end of the season, they regress those and they go out of breeding condition. They basically, birds go through, temperate breeding birds go through puberty every year, basically. You imagine that. Could you imagine if you go through puberty every year? Anyway. Yeah. And when they're breeding, they're often not, they're hanging out on territories, and they're not in big flocks, right? Because they want to protect the resources to be able to provide for their offspring. But in winter, it makes sense for them to be with other birds, because they want to go, if somebody else finds food, it's like a reciprocal altruism thing, they'll go eat with that bird, and later they'll show that bird where that food is, and so forth. So we just generally see more flocking in winter and fall. So that is going to drive, that's responsible for a few other data points down there. So those are definitely the data points. I haven't actually taken them out to see if the R squared goes from like 0.005 to 0.00005, but there's no relationship yet. But those spots are definitely those birds, so those species. Thanks for that question, Dr. Lewis. Amy, I see your hands up.
Yeah, you talked a little bit about plans to measure the canopy.
Yeah.
How do you begin to measure the canopy?
Yeah, it's tricky. So there's a couple of different ways, and there's a lot of back and forth in the literature as to what's effective and what's not. So I've got a couple of tools. I have this little densiometer, which is like a PVC pipe that looks like a periscope, basically. And you can go from the place that your you're doing the survey and you can walk, and every like so many meters, you estimate the percent canopy cover through this little periscope thing. And then you can aggregate those data to kind of a total coverage. Another way to do it is we just got some field equipment through UW-Bothell funds. So thanks again, UW-Bothell. And one of those things we got was a camera with a fisheye lens, which we can put up on a tripod and get kind of this like kind of big picture, and then do the same thing on a computer basically that we would do with our eyeballs in the field. So those are the techniques that we're going to try to do, but we haven't started yet. And we're going to need to, that's going to change seasonally as well, right? With the deciduous trees and everything. So that'll have a seasonal component as well. Thanks, Amy.
We have Carolyn. You're next.
I just have to say, I love that comment from your student. Is that Kira? It says, "Oh, that sounds so much easier." The fisheye camera.
Yeah. We when we went out with Kira and Alex and Andrea, yeah, one of the coolest parts of this, I hope Kira thought this was cool, is we would like be brainstorming consistently in the field. The students and I would sit and we'd be, initially masks on when we had to do that, and outside, and we were just we were consistently like, how are we going to measure this? We read papers and then we talk about it in the field. Then we'd be like looking up, and we'd realize like we're not always standing in exactly the same space when we're doing these surveys. How in the world are we ever going to know? We can't necessarily stake out these places because they're in public parks. So it was a lot of fun back and forth. So Kira and I kind of chatted quite a bit about how to make those measurements. So yeah,
that's awesome. And that my question, oh great, is around involvement of students and kind of what your students learned through this process and what was surprising to them, maybe about in this work, and where they go next. Where do your students go after being in your lab?
Yeah. Well, when Doug Esser talked to my students, he's in the kind of public relations, group, of publicity group here on campus. I think the things that they said were surprising were the number of species that are out there, right? As kind of like being interested in animals and animal behavior, and then suddenly seeing like, wow, there's this many species at Grace Cole Park, that really kind of was surprising to them. And then, having to learn all these little subtle sounds, like I should have played some. I mean, like the sound of like the golden crown kinglet and the brown creeper are so identical. And then we're out there like debating. We two people do the surveys, and then we face each other and say what we heard. And like the back and forth, I think it was a brown creeper because it sounded more scrapey. And then somebody be like, but it was so high in the canopy, I think it was a kinglet. And then I saw, I think that surprised them as well. As far as what they're going to do, I know Kira is, interested in animal behavior and wants to do cephalopod research, and she's still here at UW-Bothell taking classes. Alex moved on and now has a job in a lab looking at toxic shellfish algae, and they get these like mass, things of shellfish, and they have to test them. And the reason I bring that up, which I think is cool, is that, we tried to really go through the scientific process in this study. So the idea is you could take any of these skills related to bird diversity and move it, as Dr. Madzima said, move it to some other field, and you'd have the skills to be able to be successful. So I was really excited that Alex was able to get that job, partially with some of the experience he got in this project. So.
What an impact. What an impact. And this is so heartening to hear. In fact, on that canopization, I can't say that, but how we are getting it. Do you guys have drones that you can fly on top and then take an image from the top?
Yeah, I got to tell my colleagues, we got our drone in, and we have one. Yeah, it's sitting in the office now. We haven't even unpacked it yet. So I know Jeff Jensen has done some drone work as well as some other folks outside of biology, and that's something we want to do too. I mean, we have these great aerial photos, but we're really constrained because we're not taking them, right? They're from King County's archive. So yeah, we hope to use some drone footage as well.
So on sound though, you said 67% were by sound.
Yeah.
And is there like a sound database where you can actually use some now pretty easily available AI algorithms that can quickly help you to match in real-time basis, where you don't have to resolve whether it's species A versus B, because human ears, while they a lot of experience goes into it, it's hard sometimes because of you cannot hear it as well as. So maybe by combining technology, you can fly a drone, capture the sound, AI analyzes and categorizes it. Is that something?
Yeah, so that technology exists, and we use it sometimes when we're practicing. There's a lot of practicing, which is great, to go out and try to figure out what all these these bird sounds are before we do the surveys.
Yeah.
And I have one called BirdNet from Cornell Lab of Ornithology, and occasionally I'll pop it up and try to figure out what it is. The problem is that it's you're not in like a a soundproof room with the bird. There's a whole lot of other sounds, including other birds and people and anthropogenic noise and everything else. And like if there's one crow calling and you're trying to figure out what this like high-pitched contact call of a bird is, it will say American crow every time. So the technology is not good enough to kind of parse through all this soundscape and do this unless the bird is loud in front of you. It's great for like, and it's too hard when you're trying to, in the survey, you're with time trying to write every bird down you see and hear. It's too hard to also have that tech. It's great for learning, and there are repositories of recorded bird sounds we use for that as well. But that recording and the kind of machine learning thing is really effective for that, but it's not effective for on-the-fly surveys. Not yet. Maybe, in years to come, it'll get better, but it's that differentiation of sounds within a larger soundscape that makes it really difficult. We tried. I mean, that'd be easy. I'd rather sit my phone out and, a camera and then, sit inside and sleep in and not get up at 4 o'clock in the morning, but it hasn't worked yet. So.
Well, it's interesting you ask, because in the speech recognition area, especially with some of the new technology coming in, where they can literally in a room full like this, when multiple people are speaking, they can now actually identify who's speaking. There might be some. It sounds like a computer science collaboration that can happen, where you can try to build neural network models based on noisy signals. So if you can train, then the model gets better and better as you train them, and next time it will recognize the American crow and ask you, do you really want to record the crow or something like that? I don't know. My engineering brain is just lighting up. It's so fascinating.
Yeah, let's talk. No, I did some work, just really briefly. I did some work with Shima Abadi in mechanical engineering, where we were trying to differentiate crows and their location in these pre-roosting aggregations on our campus. And one problem that we ran into is when they call over the top of each other. That threw things for such a great, like if they just said caw, like it was wonderful. We could say that's a crow, it's right there. But as soon as it was overlapped, it's like all bets are off. So that, I think, is the big engineering kind of hurdle, is when these sounds overlap, is how to.
They're just like us, essentially. We all talk together.
Yeah.
So I know we're at time now, but I do have a couple quick questions. One is, when you go out in the field and you say that you record your observations, are you physically writing down your observations, or do you also video and sound record your times?
We only write them down. We don't use a voice recorder because we don't want to talk during it, and we don't record it because we would need multiple microphones.
Right. Yeah. They're directional and all. Yeah. That was a whole slew of other questions that I was not going to bore everybody else with if it didn't go. The second thing I was curious was, so I live in a rural area and I spend a lot of time out in literally the middle of nowhere, and there is so much noise and Dr. Pathak and I tried doing a video, and it was painful because of all the background noise, on campus and stuff like that. So I'm kind of curious, what kind of noises do you observe? Like, do you write them? What do you write down? Like, what noises are you listening to? What's interesting to you? Yeah,
so we always write down. I have two. Right now I'm using my telephone, but I need to get a proper SPL meter. I had one, it didn't work very well. I use 2 different apps, and it's funny, I get different measurements from them. And I take about a 3-minute sample before each survey, but it gives us a good relative measure. Then we also take qualitative measurements, and each person does it. And we may be in disagreement, and then we talk about it after the survey. Whether it's low sound, moderate sound, high sound, or loud sound, whether it's continuous, intermittent, or both, and then we write notes about it. We write how many humans we saw and how many dogs we saw within the 50-meter circle. And we have all those data. So hopefully we can combine those. But we get everything from leaf blowers to jets to people asking us what we're doing to, I mean, it's across the board. And then the birds themselves, some birds are louder than others, like the Stellar's crow. It can interfere with your ability to hear the other species that are out.
Okay, well, the amount of data that you're recording is going to be an amazing set of data. Like in 5 years, I'm excited to see what all that you guys can do with that. So,
thank you.
So as Sarah said, we are out of time, but as we can see that, this is what we want. We want a group of people who are just, talking research. And this, I think it's a fundamental part of us being humans, is that, we are by nature curious. And if we can create an environment where, we can gather together and, look into these problems. And what I really love about the fact that, we are getting exposed to this amazing diversity of research that's happening on campus. And so our goal is to promote this. To be very honest, when we read, I still remember the day I read your proposals as part of the first SRCP cycle when we received the proposals, and we were going and looking at it and saying, this is fascinating. I really want to see what happens. Today, it's a milestone day because I cannot explain how exciting this is for us. I know Carolyn shares this with me. Sarah, I know Cindy, we work on these things because we hope that, it will make a difference. And so thank you for your engagement. I know you guys are busy. I know you could have actually recorded more sounds or analyzed more data or look at it. But I think part of our job as scholars is also to share this passion and onto others. And to have students here who are actually so engaged. And what I love is that it's not about your project. It's about their education. It's about their future, their careers. We are creating future researchers, future scholars, future leaders. I think I personally could not have envisioned more from this research grant program. I'm going to write a personal note to Dr. Sharon Jones. I'm really grateful that it might be, we oftentimes shy away saying, hey, what is $250,000 going to get us? I mean, it's just such a small amount. It's not about the amount. It's about that initiative to that whatever it is, even it might be a small bit, but it can make a difference. You guys are making a difference. So thank you both, Dr. Madzima, Dr. Wacker. I cannot tell you how grateful I am to see that, we are trying to build this infrastructure. We on this campus are doing outstanding research. We don't have all the resources that our, counterparts across the lake may have. That doesn't necessarily mean that we are actually very resourceful, and we are doing top class impactful work, and we are engaging a student population that typically is underrepresented when it comes to A grade research. So I think we are making a huge impact. So thank you, and thank you to the audience for coming here, asking questions, engaging, because as you know, I'm an amateur musician, and I always say that, as much as you need trained musicians to perform, you also need audience who can appreciate that music. This is no different. Research is a community effort. We are in this together. We learn from each other. We excite each other. We ask questions to each other. When we do it right, magic happens. And thank you for such a strong start to the whole Husky series that we're going to roll out. Hopefully, we'll celebrate in person in spring in our research gala. But until then, November 17th, watch out for your mailboxes. Please come for the other talks as well, because it's fun. This is so many things that I learned today. And I actually, Doug, I have some notes on some of the statistical models that I could share. I'll reach out to you separate offline on that.
Thank you.
But this is fun stuff. So, well, I'm going to actually see if Carolyn, you want to add a line or two, and then we can end.
I just want to thank everybody so much for the participation, and especially our speakers who kicked off the series. And Surya, thank you, Dr. Pathak, for being the chair of the Research Advisory Council. You can see the enthusiasm is infectious. So thank you. Have a great evening, everyone.
Thank you, everyone. We look forward to seeing you in our next one. Have a good evening.